Okay, hello everyone. For those of you who don't know me, my name is Paul Hacker. I'm a PhD candidate in the Integrated Remote Sensing Studio in the Faculty of Forestry at UBC. And today I'm going to be presenting some of my recent work exploring variations in the accuracy of plant functional trait prediction due to spectral mixing. So just a reminder of the site that I work out of. It is the Couch and Garry Oak Preserve located on Vancouver Island or near the city of Duncan. It is owned and operated by the Nature Conservancy of Canada, and according to them, there are 164 plant species present on site. Of those 164 plant species, we've only sampled 22, and they cover a variety of life forms and families. On the left, you can see a bar graph that demonstrates the life form on the y-axis and the number of samples for each life form on the x-axis. The white numbers within the bars represent the number of species that we have for each life form. The pie chart on the right is sort of a representation of how the distribution of families from our plant composition samples, and the colors relate to the bars on the line graph. So different shades of yellow represent herb. You can see there's one red pie slice that represents the shrubs, and then two oranges represent families that contain both shrub and herb life forms. A quick reminder of how my research flow has been going. Last year, I presented on my research examining interspecific trait variation in Quercus garryana across the CGOP. And essentially, the trait variation was in relation to anthropogenic activities such as agriculture and encroaching coastal Douglas fir forest and roads. That was published earlier in 2020. Still sticking with the leaf scale analysis, but moving from intraspecific to interspecific. I'm currently looking at the effects of multiple plant species occurrence within a single pixel on functional trait estimation. This is important as my research will work towards scaling from the leaf level to airborne levels using our drone imagery. And especially in the Couch and Garry Oak Preserve, individuals of different species can occur within the same pixel. You can see from this abstract diagram below that as the pixel size increases, some of these smaller, these plants with smaller footprints can start occurring in the same pixel. So essentially, we're trying to figure out what effect does that have on trait estimation. So there are two hypotheses that I'm currently exploring. The first is that increasing presence of non-target plant species within a pixel increases the error in prediction of the traits of that target species. So the goal is of this hypothesis to observe how trait predictions change in relation to the prediction of the pure target species. The second hypothesis focuses on identifying which traits change the least due to mixing, and perhaps should therefore be utilized instead of others when trying to evaluate functional traits in mixed scenarios. So there are 8 focus traits that were selected, and they relate to leaf growth and leaf structure. I've conducted PLSR using all the samples of 21 of the species to estimate a general site model for the following 8 traits. As you can see, chlorophyll and carotenoids did not perform particularly well, whereas carbon area and the structural components lower down, LMA, LDMC, and EWT, performed better, similar to the findings Sean presented on the full CABO spectrum of spectra. So methods, this is just an overview of how I've gone about mixing, first creating the mixed spectra, and then evaluating them. So the first thing to do was select 5 different species of different life forms and compare them to a target species. And so within this analysis and in the initial results that I'll show you, you'll see a herb, 2 herbs, a shrub, and a grass, and it's all compared to the target species Visativa. The Visativa was present in every single every single field plot, and its growth strategy is quite interesting, as it is it's a legume that sort of spirals and climbs up. I wouldn't say a host plant, but it's adjacent plant species. So you could have a V. sativa wrapped around a camas species, and which would assumedly affect the spectral reflectance that you get within a pixel capturing those two. So once the species were selected, you essentially create spectra for each species spanning 10% to 100%. So the math behind it is very simple. It's just you take the reflectance band, you take that value, and you multiply it by whatever percent you're trying to get. So if it was 10%, you would take the reflectance value of band 550, multiply that by 0.1, and that is your new reflectance for V. sativa 10%. And you do that across the entire spectrum from, across the entire spectrum. So once you've created the initial percent spectra for each of the individual species, you can start combining them to create mixed spectra. And that's simply just combining one species 10% spectral variation with the target species 90% or so on. So we do that from CQUAM 10 plus VSAT 90 all the way up to VSAT 90 plus CQUAM 90 and VSAT 10. And the example you can see of the output from that is on the right-hand side in that plot. At the very top spectra in pink is the pure Camassia colmash, the CQUAM, and then you get the range of variations as we move down. And at the very bottom is the pure Vicia sativa spectra. For each of these mixed spectra, we then go and predict the traits using the coefficients that we extracted from the general species PLSR model, which I talked about on a previous slide. And then from that, we plot those in terms of their percent, which I'll show later, and determine how different the predicted traits from the created spectra are from the original predicted value of the target species. So some initial results here for a 2-species mix. You can see on the left-hand side just a line graph showing the effects of V. sativa mixing on predicted chlorophyll A. The y-axis is the percent change in chlorophyll A, so that minus 30 means that there's a 30% difference, plus 10 means that there's 10% difference. You can see overall that the performance was relatively good for most species, except for Senicula crassicalis, which had almost a 40 or a 35% change. And the accompanying bar graph is just demonstrating where the majority of points of predicted traits fall within our 10% threshold. And overall, it looks pretty good, except for the one outlier of Senicula crassicalis. So chlorophyll A, if you remember, didn't have the best model performance, and with this increasing percent change from S. crassicalis, you could draw some conclusions from that. I'll say that for the discussion. The next result that we have is carbon area. As you can see, the spread is very small. There's a very small percent change from a mix of 100% of the pure V. sativa to a mix with 50% V. sativa and C. colmash. And that's demonstrated again in the box plot, which was very interesting to find. This was the highest, this had the least amount of spread. It was the best in terms of minimizing percent change in a trait. The next is leaf mass per area. Again, a 2-species mix with V. sativa as the target species. And as we move towards a 50-50 mix with Camassia colmash, LMA performed relatively well. It was just outside our very stringent threshold of 10%. That is based off of essentially taking half of the mean trait variation for V. sativa. So for all of the traits that we measured in V. sativa, they varied approximately, exactly 20.75% from the mean. So instead of setting our thresholds at 20% because of the model area, I cut it in half to 10 to see if this would even be worthwhile to do. And yeah, again, LMA looks pretty good, except for the two species that jump out. F. idahoensis is a grass, and then again, Senicula crassicalis is showing a larger percent change as you get closer to the 50-50 mix. This one is equivalent water thickness, which was the worst performer. As you can see, the majority of traits fall outside our threshold as we get closer to the 50%, and even at 75% V sativa, you're still seeing many of the species, crassicalis, quamash, and Lamadium utriculatum, all falling either on or outside of that threshold. So EWT was not great. So coming back to the hypotheses, an increasing presence of non-target plant species within a pixel increases error in prediction of traits of the target species. Yes, error did increase as the percent of the target species decreased in a mixed pixel or in the mixed spectra that we created. L. utriculatum and V sativa trait prediction error increased the least across the traits, which is interesting. It's a herb-herb mix, so there could be more functional similarities there. Then F. idahoensis and V. sativa prediction area increased the most across traits, and that is a grass herb. Again, there could be significant functional differences causing those error increases. Hypothesis 2 was that some traits are less accurately predicted as the number of species within a pixel increases. We found that carbon area was highly transferable. As you saw in the results, there was less than a 5% change for each species, and EWT, as I mentioned, performed the worst. This table just rates each trait and ranks them based on the cumulative error at 50%. So as you can see, for the 5 species combined, the error was, or the percent difference from the pure V. sativa spectra was 12.56%, which was great, and the EWT at 220 was not great. Next steps, we're going to do 3 and 4 species mixing and apply that to hypotheses 1 and 2 and see how the increase in species or the further increase in species changes the results. And then there's another question, it's not a hypothesis yet, that we'll explore, which is what is the optimum number of species in a general model to predict traits of a non-target species? Essentially what we want to do is pick a target species, take it out of the general model, and see how well that general model predicts that trait or predicts the traits for that species. And then we'll slowly reduce the number of non-target species in the general model until we hit a threshold where the prediction accuracy is no longer acceptable, and try and determine what is the ideal number of species we need to get an accurate functional trait for all the species in CGOP. Thank you. Any questions?
Thank you very much, Paul. So we have time for one short question.
Thanks, Paul. I had a question. So,
yes, sure.
Your last question, doesn't it depend more on whether that target species of interest that's not being used to build a model is sort of within the range of trait values that were used to build a model, and not so much how many species are used? Because you can have a large number of species, but if those species are way outside in terms of the traits of that species, you're unlikely to have a good model.
Yes, that is definitely true, and that's one of the things that we are considering. If you go back towards that, or at the beginning, I had that the life form distribution, and one of the things that I had been thinking about was that if let's say you had a general model with only grasses and tried to predict a herb, it would be likely, as you say, not great, because that species is already probably functionally different than the grasses. So I definitely agree with what you're saying, and I think that perhaps ensuring that we have a spread of different life forms or other perhaps taxonomic identifiers, we could create a model that was a good enough representative of all of the variations for each of the species or life forms, and hopefully get a model that's accurate. But I do agree that there's a likelihood that question will not be approachable in the species sense alone.
Thanks, Paul.
Yeah.